All right, so when we look at this is a genomic sequence. So genomic DNA is the DNA that is in the nucleus of the cell. And I often will ask you, does genomic DNA mean that it's the DNA that is in genes and only genes? And you're supposed to say, no, genomic DNA is the DNA from the nucleus. It has genes plus DNA that does not encode any other molecule, so it does not get transcribed. So it's genes plus not genes or non-genes. So in this sequence here, I started with the chromosome, whatever, in the first nucleotide of that one chromosome. This is part of the chromosome. And we're looking at genes plus non-genes. And one of the things that people do is, I want to know where in here the genes are. So one way of looking for the genes is, I know that proteins start with methionine. So the first amino acid is a methionine because that amino acid corresponds to the ATG, meaning on the mRNA. So in here, if the mRNA is going inside of the ribosome, when the ribosome finds an ATG triplet here, which is a codon, then it says, oh, that's my first amino acid that I'll add to this chain. So the red dots are the amino acids. The one on the top was the first amino acid it added. Maybe there's more here. But the first one is the methionine, which is when the ribosome read the first ATG. So the ATG is called the start codon. And if you're like, okay, this ATG might be the beginning of my protein, but then I put a bunch of green ones here, and you're like, I don't know, there's a bunch of ATGs. Which one of these is the first methionine of that protein, and which one is just some methionine inside of the protein? Or maybe that ATG is just an ATG that does not actually encode anything. So that's the part of DNA that is not a gene. So one of the things we do is, with the program that you guys are going to run, is look for ATGs. And maybe right now you're like, hi, but Dr. T, how about, let's see, CAT? No, how about cat? And I'll be like, good for you. You reverse complemented this and you thought, maybe the gene is on the other strand and you gave me the template strand. So the computer also reverse complements and then it looks for ATG on the reverse complement of this sequence. So it's going to look for the ATGs as potential start codons. The other thing it looks for is stop codons. And let's pretend that I think that this is a gene. So this is not really the beginning of the gene. The beginning of the gene is upstream of it. But I say, oh, this is the start codon of this gene. And I go down, and if I count this every 3, let's say that this TAA is in frame to be read as a TAA. When I mean frame, I mean that I went every 3, and hopefully I'm reading here TAA. And then I'll go like, okay, the protein starts here and ends here. That's too short. That would be too few amino acids to be a protein. So the program you guys are going to run will look for ATGs. We look for stop codons, there are 3 kinds. Let's see what else. It's going to look for, is this in frame? Is this TAA? Or maybe when I went every 3, it actually reads ATT, in which case that's not a stop codon. And is this distance a reasonable distance to say that this is a protein? And the other thing the program does is look for these things, the yellow things that are marked here. These are called Tata boxes. And they are a signal for the RNA polymerase to bind and then transcribe a gene that is, or starts transcribing there, and then you have a gene downstream from it. So the gene actually would start upstream of the start codon. So the Tata box doesn't have to be in frame with this ATG. but it's a signal of start of transcription. So here is a possibility of beginning of a gene. There's another one here where you're like, oh, maybe here, this is actually too close. People do the analysis to see how far is the data box upstream of my start codon. These are too close. And the data box is for eukaryotes. Prokaryotes have their own data box. It's almost exactly the same sequence, but not quite. And but it's just think that this gene predictor software is looking for cues for where to initiate transcription, or where the RNA polymerase and the transcription proteins are going to attach to then transcribe a gene downstream. So let's see. And the other thing that I highlighted here was the stuff in blue, which are SSRs. They're often not within the gene. There are genetic diseases that are caused by a variant on the length of these SSRs. So Huntington disease is an example. Fragile X is another one, where the SSR is in the coding part of the gene, but oftentimes SSR is not inside of the gene, or if it is inside of the gene, is in a part called an intron.